Welcome to my channel Adhyan with Flow. In our previous video, we already discussed about mitosis. Now it's time to meiosis. Meiosis occurs in sexual part, sexual part, and they make in female egg cells, in male sperm cell. But mitosis division, they occur in somatic cell. They're responsible for body growth and they are responsible for egg formation and sperm formation. Clear, guys? Now, meiosis divided into two parts. Meiosis, this division is not a single step division like prophase, anaphase, metaphase, anaphase, telophase. No, they are meiosis 1 and meiosis 2. They are occur in meiosis 1 and meiosis 2, and they follow this prophase, metaphase, anaphase, telophase, as well as meiosis 2 is same to same is mitosis. Their step, their functioning as it is as a mitosis. Okay, now meiosis involve two sequential cycles of nuclear and cell division called meiosis 1 and meiosis 2, but only a single cycle of DNA. Okay, yes guys, we know that interphase in our previous video already discussed. M-phase, here this process meiosis 1 follow both the process, but meiosis 2 only follow M-phase, because when this cell divide and they goes hand to hand in this pathway, when single cell divided into two cell, these two cell instantly goes in meiosis 2, so it does not take a time for interphase G1 as Z0, 0 or Z2. Okay, so meiosis 1, yes, they have S phase, they have Z1 phase, they have Z2 phase, but meiosis 2 does not have. Okay, now let's start with meiosis first. First step is prophase. Like same guys, these are nuclear envelope and this is our cell. Clear guys? Now let's move further. Prophase of the first meiotic division is typically longer and more complex when compared to prophase of mitosis. They say that meiosis is tougher compared than mitosis. Prophase, this stage take more time because they divided into 5 phases also: lactotine, zygotine, pectine, diplotine, and dikinesis. Remember this sequence guys: L Z P D D I. Lata zara pani de de. Okay, forget. Okay, forget. Lactotine, zygotine, pectine, diplotine, dikinesis. Let's start with lactotine, our first phase of the prophase first. In this stage, chromosomes become gradually visible under the light microscope. Important guys. The compaction of chromosome continues throughout lactotine. This is followed by the second stage of prophase. They want to say that here is chromatin condensation start. We can also say compaction of chromosome in this stage. That's it. Next, what happened in next? Again I'm drawing, this is the nuclear envelope and this is the cell. Zygote. During this stage, chromosomes start pairing together and this process of association is called synapses. Such paired chromosomes are called homologous chromosome. Electron micrograph of this stage indicate that chromosome synapses is accompanied by the formation of complex. We can call it as a synaptonemal complex. Now listen to me guys. For second stage, now listen to me. They said that here homologous chromosome come closer guys, with the help of synaptonemal complex. This is the synaptonemal complex. And what is these? Homologous. What? Homologous? What? Chromosome. Clear? What is synaptonemal complex, guys? They are made up with lots of enzymes, lots of protein. I'm saying again, they are made up with lots of protein and some kind of enzymes. So both the homologous chromosome attached tightly, compact. So we can say it as a synaptonemal complex. Now I'm reading further. The complex formed by a pair synapsed homologous chromosome is called bivalent or tetravalent. If I concentrate only this part, look at my pen, this chromosome, look at this chromosome. If I consider these two chromosomes, then we can say bivalent. If I consider whole, then I must to say tetrad. Now you clear, guys? Look at me here. This is the guys, what? This is the bivalent. And this complete is tetravalent. Now let's move further. Chitin, very important, guys. This is the crossing over phase. Important, guys. This is the crossing over stage. In which stage crossing over is occurring? Now you have to remember that this is the meiosis first. Prophase first, and their third stage, chitin. What happened in crossing over? This is the exchanges of some genetic material. You know, now one chromosome comes from your father side. This chromosome is occurring from, provide from your mother side. Now we have to exchange the gene. So you can get both kind of genes, because this structure goes into you. Now guys, what happened with this? Now their chromosomes arm, they combine together. I'm not making here synaptonemal complex, I'm just drawing how they combine. This here is this like arrangement. This is for another chromosome and this is for another, and this cross form we call it as a chiasmata. Clear, guys? Now you clear, I know you are clear. Okay, let's read, guys. This is the chitin, guys. This is the chitin stage. Now let's read. During this stage, the four chromatids of each bivalent chromosome become distinct and clearly appear as a tetrad. Yes, guys, this stage is characterized by the appearance of recombination nodule, the site at which crossing over occur between non-sister. Yes, guys, this strand and this strand is sister, but this strand and this strand is non-sister. So crossing between non-sister chromatids. This is the one chromatid of different chromosome. This is the one chromatid of different chromosome of the homologous, but they are same chromosome. Crossing over is the exchange of genetic material between two homologous chromosomes. Okay, I already told you. Crossing over is also an enzyme mediated process, and enzyme involved is called recombinase. This is not a simple process that they come together and connect with them. No, there are lots of enzymes are required. We call it as a recombinase. So they recombine both the strand. So they recombine all the strand, all the only homologous chromosomes, non-sister arm. Crossing over leads to recombination of genetic material on the two chromosomes. Recombination between homologous chromosome is completed by the end of chitin, leaving the chromosome linked site of crossing over. Now you are clear what happening in this is the stage of crossing over. They can attach and cross their self. This is the node. We can call it as a chiasmata. Now next step is this is the diplotin. Now this comes to diplotin. Now in this stage we can betterly watch what chiasmata. This stage that chiasmata is formed, but in this phase chiasmata actually visible properly, because this is the starting phase and this is the end of chiasmata. Diplotin is recognized by the dissolution of the synaptonymal complex and the tendency of recombine of homologous chromosome of bivalent separate from the each other except the crossing over side. And now let's see, guys. They want to say that now what happened? What happened in diplotin stage? What happened in diplotin stage? That synaptonymal complex, they get dissolved. They get dissolved. They get dissolved, and both the separation attach will finish. Both the separation and attachment is dissolved. So look at me here, read paragraph. The tendency of recombined homologous chromosome of the bivalent separate from each other. So these bivalent separate from each other, but they are not released at a specific site. They are not released, but this bivalent, now they are attached. Now here they are attached, here they are attached, but now they don't attach, and we can easily visualize what chiasmata formation. In oocytes of some vertebrates, diplotin can last for months or years. Okay, ठीक है. The final stage, meiotic, means meiosis, guys, don't be confused. Dikinesis. This marked by terminalization of chiasmata. During this phase, the chromosomes are fully condensed. Okay, they are fully condensed. First thing is they are fully condensed. The meiosis spindle is assembled to prepare the homologous chromosome for separation. By the end of dikinesis, the nucleolus disappears and nuclear envelope also breaks down. Dikinesis represents transition of metaphase. They want to say that here what happened? Now spindle fiber become arise. Nucleolus disappear. Nuclear membrane start disappearing. Look at me now, guys. Now spindle fiber also form. Spindle fiber form, and now there is terminalization. Now at the end of diplotin, this chiasmata start disappearing. This chiasmata start disappearing. The crossing over is end. They crossing over is completed. They exchange their genetic material in the dikinesis stage. Now this chromosomer, they attach with spindle fiber. They attach with spindle fiber. Clear, guys? And this phase now enter into metaphase second. In metaphase second, what happened? The bivalent chromosome align on an equatorial plate. Now they align on an equatorial plate in metaphase. Let's check me out. Yes, guys, look at here. Look at here, guys. This is the prophase. They already complete their 5-step: lactotin, zygotin, pacitin, diplotin, dikinesis. And dikinesis stage enter in metaphase 1. And now this chromosomal arranged in an equatorial plane. This is the equatorial plane. Equatorial plane. They are attached with spindle fiber. Look at me, look like here. And this is the kinetochore. And spindle fiber attached at kinetochore. And their end part is connected with where? They are connected with centriole. Now, what happened after metaphase? The homologous chromosome separated, split, now split, while sister chromatid remain associated at their centromeres. They want to say that this is the homologous, and this part, this part move this side, and remaining part move this side. They still attach with spindle fiber and move towards their different pole. They split. Now this structure is formed. Now they split. They split. Look at here, guys. Now they split like this manner. Okay, clear. Now you can clear now, guys. Now you can clear betterly. And now finally nuclear membrane start forming telophase. I'm drawing here telophase. Now this is the, wait guys, not cytokinesis still occur. Here nucleus is formed, karyokinesis is occur. In the cytokinesis, one cell divided into two cell, one cell divided into two cell guys, and this cell further enter in meiosis second, and this cell further enter in meiosis second. Look at me. Telophase, the nuclear membrane and nucleus reappear. Yes, reappear. Cytokinesis follow, and this call it dyad of cells. Although in many cases the chromosome do undergo some depression, they do not reach the extremely extended state of the interphase nucleus. They direct enter in meiosis second. They don't touch interphase. This cell don't touch interphase. They directly enter in meiosis second. Let's start with meiosis second, guys. Let's start with, and these are simple. This is the simple, guys. This is simple. This is same to same according to mitotic. This is same to same according to mitotic. Now let's start with prophase. Meiosis second is initiated immediately after cytokinesis. Okay, usually before the chromosome have fully elongated. The contract of meiosis 1, meiosis 2 resemble a normal mitosis. The nuclear membrane disappear. This is the fast, guys. Now what happened? I'm taking this one cell. I'm taking only one cell. Now nuclear membrane start dissolving. These are chromatin. They start compaction. Okay, look at here, compact. Now what happened in metaphase? What happened in metaphase? At a stage of chromosome aligned to the equator. Same guys, same now, same again, same. This is the cell. Recently nuclear membrane, nuclear envelope dissolve. Here centriole reach to there. You have to write in your exam. There rise spindle fiber. Here chromosomal align in an equatorial plate. At an equatorial plate they are connected with spindle fiber. Now they connected with spindle fiber. Clear, guys? Now what happened with them? This phase enter in anaphase. This phase enter in anaphase. It begins with the simultaneously splitting of the centromere of each chromosome, which the holding the sister chromatic together, although they move towards opposite poles. Now what happened? They now split. Splitting is start. I'm making this kind of centriole. Now what happened? Anaphase, the sister chromatid becomes separate. This set going towards, and this set going towards this side. Clear, guys? Now okay, wow, look at the diagram. Look at the diagram. In prophase second, they are nuclear membrane is, I'm taking only one cell. This is the another cell. Okay, they attach with the spindle fiber, guys. They attach with spindle fiber. Now in metaphase they arrange in equatorial plate. They arrange in equatorial plate. In anaphase again they split. They split properly. And in telophase nuclear membrane become reappear. Okay, nucleolus become reappear. Now they form, this single cell form 2 and this cell form 2. This is also attached, guys. This is not a cytokinesis stage. This cell is attached. This cell is attached. After cytokinesis they become separate. So at a starting, when we start meiosis first, we get two cell. At the starting of meiosis 1 we have a one cell, and they split and divide into two cell, and these two cell enter in meiosis second, and now at the meiosis second ending we got four cell. Okay, guys? Now what is telophase? The meiosis end with telophase second, in which the two group of chromosome once again get enclosed by nuclear envelope. Cytokinesis follow, resulting in the formation of tetrad of the cells for 4 haploid total. NC arrangement. NC arrangement. Now what is the significance of meiosis? What is the significance? They occur in our reproductive part. They occur in our reproductive part. That's it. Now they passes generation to generation. They passes generation to generation because the most important phenomena occurring in meiosis, crossing over which stage? Which stage? Pachyty. Bracket prophase fast. Okay, because variations are very important for the process of evolution. If you don't have any kind of variation, if you don't have any kind of feature from your mother and father, then you can't adapt with nature. So this is all about your meiosis. So that's all for now, and thanks for watching me. Thanks a lot, guys, and please subscribe my channel. That's all for now. And revise, guys. You have to revise in which stage what phenomena is occurring, in which stage what phenomena occur. You have to learn properly. If someone asks, like, chiasmata betterly visualize in which stage, and you have to remember, yes, they can betterly visualize in diplotin stage. What is the termination of chiasmata? And you have to remember that, yes, okay, dikinase is the termination of chiasmata. So you have to remember, guys, you have to remember this cell division in your finger marks, which stage, after which stage, what is the stage when equatorial plate formation, when a chromosome align in an equatorial phase. So you have to remember in which phase what kind of chromosome ratio you have to remember. Okay, so thank you, guys, thank you so much.